a revision stream. It has been a while since I've done one of these, purely because I was ill all of last fortnight and then some of last week, and obviously last week was the holidays. So, I have actually missed quite a lot. I have missed quite a lot of content, and what I'm going to try and attempt to do in today's stream is catch up as best I can. I'm going to go over things like insulin, adrenaline, which completely blank on. Like, I have no idea anything about that apart from that it's secreted from the adrenal glands, which I don't even know where the adrenal glands are. That's how, like, non-existent my knowledge is. We'll go through blood glucose regulation. We might go back over thermoregulation too. And finally, we'll start to look at the kidneys and diabetes. That's an important one, because I was looking at some exam questions on diabetes, and it's a very extensive topic. Very extensive. So, yeah. But I feel bad rubbing these out, because purged through these, but we're fine. Okay. So let's start with blood glucose regulation. And this occurs via the action of insulin and glucagon, which are two hormones. Now I'm pretty sure both of them are secreted from the pancreas, but bear with me, I might be wrong. Now what insulin does is it enables the conversion of glucose to glucagon. So glucose, as we know, is the sugar. Glucagon is basically a storage molecule used in animal cells. It has a very large surface area to enable fast hydrolysis of this molecule, a breakdown into glucose. So in other words, it's converting glucose into its storage form, its compact, highly branched storage form. From there, this is called glycogenesis. That's what it's called. And this is detected, a fall in blood glucose concentration is detected by alpha cells, which I think are in the liver. They might be in the hepatic vein and artery, perhaps. Hepatic is the word you want to use here when referring to the veins and arteries, or the blood vessels overall, the hepatic. So the fall in blood glucose concentration is detected by alpha cells. And obviously that leads to glycogenesis. So the way I remember it is really simple. I just remember alpha fall, beta rise. That's literally the way I remember it, because a rise in blood glucose concentration is detected by beta cells. And that converts glycogen into... Sorry, I wrote glucagon there. Which, I'll just be a sec. Okay, sorry about that. So yeah, glycogen is converted into glucose, so it's literally the opposite of insulin. So we have this highly branched, as I say, storage molecule, which is then converted back into the penta sugar. So that is called glycolysis. That is the word you would use. Now, there is finally one more important little phrase you need to know. It is called gluconeogenesis. And in summary, this is literally just converting non-carbs, non-carbohydrate molecules into glucose. Because if we have a fall of oh my god, if we have a fall in our blood glucose, you know, we barely have any glucose in our blood, we are going to need some. Obviously, you need glucose to be able to respire, etc., etc., for ATP production, for all of your metabolic processes. So, non-carbohydrates. And this links into anaerobic respiration. Because what you have is, and I need to go back over this because I don't really remember it that well, but I know it's fatty acids, I think, can get converted into glucose, and I think lipids can too. So you know what? I'm going to add to this, and I'm going to quickly pull out one of my old books and see if I can remember. See if I can remember. Right here. Respiratory substrates. Lipids are hydrolyzed into glycerol. Glycerol becomes triose phosphate, which is used in glycolysis. Okay. Let me write that down. So, fatty acids. Fatty acids link reaction. And glycerol becomes triose phosphate. For glycolysis. So really, you could literally just say that glycerol is used in glycolysis, fatty acids used in the link reaction. So actually, I'm going to reword this, just so it's easier to understand. Glycolysis, free carbon compound, slash triose phosphate. Because yeah, it gets broken into phosphorylated glucose, and then it becomes 2-triose phosphate, which become reduced, and then, oh god, I need to revise for all sorts, bro, oh my god, okay. And then anyways, proteins become amino acids, they enter the respiratory pathway as pyruvate or Krebs. So all I'm going to put there is amino acids, Krebs, or Okay, I'm gonna very quickly also link this to the Krebs cycle, etc. Glucose. We're gonna link this in. We have a 6-carbon compound which gets phosphorylized via ADP plus PI. And then that obviously becomes free pyruvate. That's gonna be glycolysis. So, I think that is everything for that topic. Oh, just gonna try and take a screenshot. Hopefully that's good. So, I think we're fine. I think that is good for blood glucose. Alpha cells detect a fall, beta cells detect a rise. Glycogen to glucose is... That's why I'm spelling it wrong. Glycolysis. Feel like that's the wrong word. No, it can't be glycolysis. Glycolysis, yeah. It can't be glycolysis. There's a different word there. And I'm gonna find out what it is. But, there we go. That is a good, quick bit of knowledge on respiratory substrates. Lipids, the fatty acids are used in the link reaction, and glycerol is used in glycolysis. Amino acids are used in the Krebs cycle or in glycolysis where they become pyruvate. Once they are removed of their base. Just give me 1 sec. Sorry, I was expecting someone to message me. Right, let's take a big look. It is blood glucose regulation. And we're also going to go look at insulin and adrenaline because I was not in when we did that, so I'll figure out what that's about. Very quickly though, those words. Glycogenolysis. Okay. So I literally missed a single part of it. Okay. That is now 100% accurate. Blood glucose regulation. Insulin or glucagon. And it's using enzymes. So the whole thing to really wrap your head around here is the fact that, yeah, these are hormones, but the way they achieve what they do is by activating certain liver enzymes, which will then go about and break down, glycogen into glucose or convert glucose to glycogen, non-carbs to glucose. That is the main idea you need to get around. It activates the liver enzymes. And it looks like we need to go and look at insulin and adrenaline. So, effective insulin on the glucose permeability of cells. So, I'm guessing, just from inferring that, it means insulin will make cells... I'm guessing if it's a fall, it would make Oh, that's kind of weird. Would it make cells more permeable to glucose if it wanted glucose? Sorry, I've also realised I've got these the wrong way around. That's why I was confused. Have I? No, fuck off, hold on. Sorry, have I? Alpha cells detect a fall. Oh, which is then glucagon. Okay, so I've got these the wrong way around. That's where I went wrong. A rise detected by beta cells. A fall detected. That was my mistake, guys. Again, I have not done this topic in class. I'm having to learn it myself. I'm obviously going to make some mistakes. That is fair enough, though. That makes it make a lot more sense. So yeah, you get a massive. Okay. I'll draw like a little graph, right? 2 graphs. We get a rise. So this is for normal. We get a rise. It obviously is going to need to be brought back down to normal. We get a fall. It's going to need to be brought back to normal. The way this is achieved is if there is a rise, it needs to fall. The rise is detected by beta cells. There it's detected by alpha cells. This is achieved by converting glucose to glycogen. This is occurred by converting glycogen to glucose. Easy. So just really you can just remember those 2 graphs. What must go up must come down. What must go down must come back up. That's basically the easiest way to remember it. So, if insulin is only triggered when it's a rise, would that make cells less permeable? Or would it make cells more permeable in order to draw in the glucose to prevent an increased concentration? I'm going to say that insulin probably makes cells more permeable. But let's have a look at what the PowerPoint says. Again, learning this for the very first time. Gonna have to learn. So, insulin binds to receptors on the cell surface membrane, okay. Intracellular, so it sends a chemical signal once bound. It increases glucose permeability because vesicles containing a carrier will fuse with the cell surface membrane. Okay, I think that's easy enough to draw. We just draw like a little receptor here. So what we're saying is insulin, which we're just going to draw as this. Insulin is going to bind. That is going to send a signal to the vesicles. And these vesicles contain a carrier and this carrier fuses with the membrane, which increases permeability. That is pretty easy to understand. Okay. So how does insulin reduce blood glucose level? When insulin binds to receptors on the cell surface membrane, it causes glucose channels to open, allowing glucose to enter via facilitated diffusion. Okay, so we also need to include this. I guess we'll put different examples, because it seems like it wants us to talk about multiple here. So if insulin binds, it's going to cause this to open, allowing glucose to enter. There is a rise in the number of glucose channels present in cell surface membranes. Enzymes are active. So another thing is just going to be that insulin activates enzymes. Activates liver enzymes. So the concentration of glucose is lowered because more glucose is being absorbed by the cell. Cells increase their respiratory rate and therefore uses glucose. So yeah, I guess I'll add that. Uses excess glucose in respiration. Increased rate of glycogenesis. And they activate cells which lead to gluconeogenesis. And they work via a second messenger model. So adrenaline will bind to a receptor. The protein channel changes shape and activates adenyl cyclase. Adenyl cyclase converts ATP to CAMP. CAMP activates protein kinase. Protein kinase converts. Okay, that is going to be very important. And that's everything from that topic. Okay, so let's look at this firstly, and then we can move on. So, there are 3 ways in which insulin causes a fall in blood glucose concentration. The first is when it binds, when the insulin receptor binds to the membrane, it will cause an increase in the cell surface membrane's permeability to glucose. And the way it does this is carrier-containing vesicles will fuse with the membrane. This carrier will increase the permeability. Another way is when it binds, it will open up glucose channels. Glucose will then move via facilitated diffusion into the cells. And obviously the glucose is used in respiration. But the main one is insulin will activate liver enzymes which convert glucagon to glycogen via glycogenesis. And this is the second messenger model. So the way this works is, I'm just going to see if I can quickly remember. So adrenaline is going to bind to the receptor. Protein changes shape. Changes internal shape. That leads to changes shape on the inside of membrane and activates adenyl cyclase, which I've never heard of before, to be fair. Adenyl cyclase is what converts ATP into CAMP and CAMP is what then activates protein kinase, and that is what's used to convert glucose to glycogen. Sorry, glycogen to glucose. Yes, because remember, adrenaline is released during periods of stress, excitement, okay, when we will need an increased respiratory demand. If you get into a fight, you're going to have an increase of adrenaline because you are going to need that excess respiration for muscle contraction. An increase in ATP, and that is all caused from adrenaline activating adenyl cyclase, converting ATP to CAMP activating protein kinase, converting. Okay. Well. I am going to very quickly write this down. That's what I'm going to spend 5 minutes doing. Just going to quickly write this down. This is the, what was this called? The 2 something model? Second messenger model. So glucagon, which remember, when alpha cells detect a fall, that is going to be when glycogen is converted into binds to the cell surface receptors. And it causes it to change shape inside the membrane. And what was it called? Adenyl cyclase. Adenyl cyclase. So that's an important one to remember. Which converts ATP into CAMP. CAMP activates protein kinase. Which is glycogenesis. Okay, that is honestly pretty damn easy. That is honestly pretty fairly easy, I would say. So all the adrenaline does is it binds to the receptor, causing the activation of adenyl cyclase. Adenyl cyclase converts ATP to CAMP. CAMP activates protein kinase, which is literally just glycogenesis. No, sorry, that would be glyconeolysis. Because we're not creating glycogen. Glyconeolysis. Yeah. Glyconeolysis. Let's have a quick check. Glycogenolysis, sorry, not neo. Glycogenolysis. Okay. So, I honestly think we might wrap things up there for today, purely because, that has honestly been quite a lot to go through. I know we've only been live for half an hour, but that has been quite a lot of content that we've gone through there. We've done the second messenger model, which I guess second messenger might help understand this, because yeah, it's adrenaline, and then it's adenocyclase, it's CAMP, and then it's protein kinase. It's 4 different things here. Adrenaline causes the activation of this, CAMP causes the activation of this. Second messenger, it's a messenger patting the back of another messenger that then does the job that it needs to do. So I think we could wrap things up there for today, as long as we tomorrow, which again might be a bit difficult because Purge will be at mine until probably 8. So you know what? I might wrap up the stream, take like a 10-20 minute break, half an hour, whatever, and then come back, and then I might continue this. I think I just need a couple of minutes to clear myself. We've been live for half an hour. So, insulin, adrenaline, and glucagon, this has been this part of the stream. Next, we'll go over diabetes, and then that's where we'll wrap things up. We'll do some exam questions. Ciao for now.